such a mode to make the microneedle array patch, the silicone mode, and we mix the formulation together with the hyaluronic acid solution, and then pour it to the mold. After drying, sometimes with the photopolymerization, and we can obtain such like a uniform structure. And once I talk about the microneedle array patch, always like showing this picture, you can see the setting point. Actually, it's not this guy, actually the second one. Because he's the second one waiting the line. Then very nervous, right? Other people still like smiling, laughing, and potentially with the microneedle array patch, you don't have to worry about you will be the second one. Okay, you can easily put the patch to your belly, to your arm, and quickly reduce the blood sugar level. So then we first tried on the mice, and you can see they can easily penetrate to the skin of the mice and quickly reduce blood sugar level from a high level over 500 milligram per deciliter to a normal range below 200 milligram per deciliter, and it can maintain there for a while, then gradually increase. However, if you just put the native insulin in the patch, just due to the passive release of the insulin, the blood sugar level can quickly go down and quickly go up without the such glucose sensitive control. Okay, so you can see the difference between the orange line and the black line. So meanwhile, we can adjust the insulin level as well as the glucose sensing factor amounts to potentially achieve the personalized medication. And we also performed such study. We put one patch on, the blood sugar level went down. And at this time point, so once blood sugar level already low, and then if you put a second patch, if the second patch is glucose responsive insulin patch, and you can see the blood sugar level can still like maintain around like 200 milligram per deciliter for a while, then gradually increase. However, if the second patch is just like a native insulin-based patch, and then due to the passive release of the insulin, the blood sugar level can further go down, even to achieve the range of the hypoglycemia, which is very dangerous. So then in our lab, actually, we perform, different studies associated with different formulations, different mechanisms of glucose responsive. And we are trying to identify the best one and then to push it forward, especially to potentially translate it. And this time, here's another one, and we want to mimic the fusion process of the insulin granules toward the cell membrane of the beta cells. And you can see here it's like a very elegant process of the real beta cells. They have many this kind of granules with the insulin encapsulated inside. And once blood sugar level goes high, through such like a complicated process, signaling reactions, and then eventually the calcium ion channel can be open, and the calcium ion can go inside, and the calcium ion actually can bind to the snare protein of the insulin granules to change the conformation and allow it to further interact with another two snare proteins associated with the cell membrane, and then to promote the fusion process to release the insulin. So this is really, we put like an elegant design by nature. And we are trying to mimic such process through such limited tools in our toolbox. And especially we design such like a vesicle based design. And you can see here the smaller vesicle made by the liposome, and we have the insulin encapsulated. And the larger one is also made by the liposome with a relatively larger size, typically like several micrometer. And we also anchor a peptide K to the smaller one, the peptide E to the larger one. And the peptide K and the peptide E, they can interact with each other to form such a quarter-course structure, then to promote the fusion process to release insulin. But we don't want to like allow it to happen like anytime. We really want to follow such glucose responsive manner. Then we further add a PEG molecule, a polymer to the smaller one, especially through a single-strand DNA. And this single-strand DNA can have a complementary structure toward another single strand DNA already anchored on surface of the smaller liposome. And then, due to the existence of such a PEG molecule, the peptide K can be hidden inside, and it won't interact with the peptide E immediately. However, such DNA structure is like a pH sensitive. So once glucose level goes high, glucose coming, and due to the enzymatic reaction, the pH level quickly reduce, and then allow the conformation change of such DNA structure from such a helix structure to be more 3D based structure, just like a multiple or like G contriplex. And then allow it to such a PG molecule can be detached from the surface of the smaller one and allow the peptide K to interact with the peptide E to promote the release of the insulin, actually to promote the the fusion process. So here you can see inside the smaller one, inside larger vesicle, we have this kind of smaller insulin liposomes, and they can respond to the high blood sugar level to quickly reduce the to release the insulin, and the glucose level less insulin released. Here, I mean, the difference of the insulin release rate can be 8 to 10 times difference. And in our lab, we are also trying to directly make the insulin itself responsive. So, for example, we conjugated the glucose transporter inhibitor to the insulin to form such I-insulin, okay? And you can see here, the insulin can bind to the insulin receptor to reduce blood sugar level, and the glucose transporter inhibitor actually can interact with the glucose transporter to block the glucose transporter. And if you directly inject such glucose transporter inhibitor to the body, actually can increase blood sugar level quickly. So we link them together, and then through such injection, like a sub-Q injection, or like transdermal delivery of such insulin toward the body through the microneedle array patch, they can form such like a balance toward the cells. They can either bind to the insulin receptor or to the glucose transporter. And then once blood sugar level goes high, the glucose can competitively enter the
Zhen, we can't hear you.
Hello?
Yes.
Okay, now you can hear me.
Yes, yes, go ahead, please.
Okay. Yeah, it's like the internet is not that stable. Okay. All right. So we just talked about the modified insulin, which can achieve the glucose sensitive insulin response. And once we overdose, like inject such insulin to the body, actually we found that they won't cause the hypoglycemia. And somehow it's like the balance more to the glucose transporter. And you can see here, if you inject twice of the native insulin, and can quickly achieve the hypoglycemia. However, if you inject such like insulin, which is like glucose sensitive, they won't cause a hypoglycemia. Actually, can maintain the blood sugar level around like 100 milligram per deciliter for a while, then increase. So this is like a kind of the advantage of such glucose sensitive insulin. And then we further like summarize the translation criteria of the microneedle patch. And you know, several things should be kept in mind, especially how to achieve the high loading capacity with the high bioavailability, but still like with a small size of the patch. And the biocompatibility certainly is also very important. And also associated with manufacturing should be fast, should be available for the large scale manufacturing and with a low cost. So around also 6 years ago, we set up a startup company called Xenomics and trying to translate such technology. And this is our, the milestone of the translation, especially currently we are working with the FDA for application of the phase 1 clinical trial. And now it's a quarter size, the smart insulin patch could effectively regulate the blood sugar levels of the 25 kilogram type 1 diabetic pigs in a normal range for up to like 24 hours. And this is our design. And then we identify a mixture of the monomers, actually 2 monomers with such amine group. And they show actually high positive charge on the neutral pH. And then with a strong affinity toward the insulin, which is like a negative charge on the neutral pH. And one monomer is like with 2 double bonds. So they can form such cross-linked network. And one monomer is like with a phenylboric acid. The glucose can bind to it. And then the charge will be from like a neutral charge to more negative charge. So once the glucose coming, especially with the high glucose level, and the positive charge, overall positive charge can be reduced. Then the affinity between the insulin and the matrix of the microneedle patch can be reduced and to promote the release of insulin outside. And then we can actually form such a mixture together with the monomers and insulin and simply pour it to the mold and shine the light to it. Suddenly we have the photoinitiate inside, and then can quickly form the microneedle patch. Now since like the whole matrix is like a glucose responsive, so then the bioavailability of the insulin can be significantly increased compared to our previous design, like to add such a formulation to the microneedle patch. And now we can just simply pour the mixture of the insulin together with the monomers to it and shine light to it, and it can form the glucose responsive matrix with the insulin encapsulated inside. And also, you can stop such treatment at any time. So if people feel uncomfortable, you can just detach the patch and throw it away, and further enhance the safety of the patch. And the whole manufacturing process also becomes relatively simple. So we tested on the pig, and one patch after 2 feedings, blood sugar level went up. However, for the patch without the glucose responsive control, 1 patch on, and after 1 feeding, blood sugar level went up because without such glucose responsive control. And you can continuously apply patch. Here's like to the pig, 1 patch on after 2 feedings. Second day, a new patch on, and then can last a much longer time, like over 40 hours. And after usage, you can remove the patch. And since the matrix is cross-linked, and only insulin can be delivered to the body, so the biocompatibility is also pretty good, and it just shows very limited information toward the skin. And we also perform the glucose tolerance test. So once the brush level becomes normal, we inject a bolus of glucose to the peak, and with the increase of brush level, you can see the insulin level also quickly increased. Very clear, like glucose responsive behavior. And here, like 1, 2, 3, 3 different peaks, and showing the similar trends. And meanwhile, we thought about that this could be a broader platform technology, since you can apply the different patches with different drug, and they can respond to the different physiological signals associated with different diseases, and to release the drug like locally to the skin, or through like lympho circulation, or like capillaries to the whole body to achieve the systemic treatment efficacy. So based on this, we also put the glucagon to the patch. They can respond to the high insulin level. And also recently, we can make it like to directly respond to the low brush level to release the glucagon, and quickly, increase brush level to further reduce the risk of the hypoglycemia. To enhance the safety of such patch, we even design such dual self-regulated glucose responsive patch. You can see the red part actually mimic the function of the beta cells to release the insulin once the brush level goes high. And the blue part actually can respond to the low brush level. And once brush level becomes low, more glucagon can be released to increase brush level. And here we just leverage the different PI value of the insulin and the glucagon to achieve such different functions. And also smart cell patch, we can even put the cells to the patch for the different applications. Heparin patch, which can respond to the thrombin level and to release the blood thinner. Here's like a heparin to reduce the risk of the stroke. Anti-acne patch, we can change the shape of the microneedle patch to wrap it around like a swab, and it can simply destroy the pimple or acne, and meanwhile can potentially also release the drug. We can also put the drugs, or even Hnsulin, which can promote the hair regrowth, to the patch. And here is we apply the keratin, such protein made from the hairs, as the major matrix material of the patch to release such drug, as well as the Hnsulin to promote the hair regrowth. And also through such a minimally invasive surgery to put the patch on the heart. And here's to promote the myocardial infarction, this kind of treating, okay. And also we can put the patch on the leaves of the plants. And here's for extracting the plasmid DNA, and for simple detection. And even can combine the smartphone technology for the quick PCR detection. Right? And this is our latest study, and we can put this kind of bioresponsive reaction. This case like a catalytic, to the microneedle patch. And here is a PD nanoparticle, which can convert the pro drug to the rare drug. Here's the doxorubicin. And the PD nanoparticles actually show certain toxicity, and actually somehow limit the application of the PD nanoparticles in vivo. Then we can put such PD nanoparticles to the titanium oxide nanosheets, and then to immobilize such PD nanoparticles in the microneedle patch. And then we can insert such microneedle patch to the tumor, and certainly you can also apply some other devices even to the tumors inside the body. And then you can perform the systemic injection of the prodrug, right? And here, only once the drug like diffuse into the tumors and to interact with here's the PD nanoparticles, and then the prodrug can become like the real drug. And then that means you can continuously generate the drug into the tumor and to boost this kind of treatment potential. And after usage, you can just remove the patch, right, and reduce the toxicity. And for cancer treatment, you can also put this kind of antibody or cytokines associated with the immunotherapy into the microneedle array patch, and to locally enhance the treatment efficacy. Especially, you can enhance the retention of the therapeutics inside the tumor, for example, associated with melanoma, and then to also potentially reduce the systemic dosage, okay, and to alleviate the toxicity, the side effects. And you can also change the shape of the needles, relatively. Here's like you can make such a hollow structured microneedle array patch, and even to guide this kind of cool atmospheric plasma into the hollow structure and directly associated with the tumor sites. And then the plasma actually can generate the ROS and RNS, and then to induce the immunogenic cell deaths. And together, works with the antibody released from the shear part of the microneedle array patch to boost the treatment efficacy. So besides the microneedle array patch, as I just mentioned, we are also very interested in cell therapy, using cells as carrier to deliver drug, especially interested in using the platelets as a carrier. So especially to leverage the natural property of the platelets toward this kind of bleeding sites, the inflammation sites, and we can bring the drug to the certain disease sites. And especially we can add such like antibodies, just like the PD-L1 antibody to the surface of the platelets. And then during surgery, once people remove the primary tumor, and then suddenly, cause the bleeding, and then we can infuse the platelets with the drug to the body, and then they can accumulate in the surgical sites, and then gradually release the drug. Especially the platelets can be activated to form the platelet-derived microparticles to boost the release of the drug. And meanwhile, during such activation process, the cytokine, chemokine can be released, right? And then to further help the recruitment of the T cells. So in vivo, we can say such platelets with drug can accumulate in the surgical sites, and to alleviate the tumor recurrence after surgery. Here's like associated with B16F10 melanoma, which is very aggressive, and also can inhibit the metastasis. Here's like associated with the lung tissues. And in our latest study, we want to further boost this kind of targeting capability of the platelets toward the metastasis. And then we perform such like combination treatment. First, we can inject such a small molecule, just like the valimandin. And the valimandin actually can disrupt the newly generated vessels, okay? Especially, based on the growth of the metastasis, the vessels can be generated. And then, once you apply the valimandin to it, and then it can cause this kind of bleeding associated with the metastasis. So then you further inject the platelets with the drug, here's like a PD-L1 antibody again, and then we found that we can effectively target to the metastasis sites, and then to inhibit the tumor development and to enhance the survival of the mice. And meanwhile, you can also apply the local treatment. So, for example, you can form such like a scaffold, just like lyophilized by the hydrogel made by the hyaluronic acid. And once you want to apply it, you can load the like a platelets with drug or together with these kind of T cells to the scaffold, and then to fuse such like a scaffold with cells to the surgical sites, right? And then certainly such scaffold can serve as a certain support to support the tissue, especially associated with some like surrounding soft tissues, for example, like a brain, and then the cells can gradually release from scaffold to boost the treatment efficacy, and the platelets can run a bit fast, and they can stop bleeding, and meanwhile the drug can be released just like a PD-L1 antibody, and meanwhile the CAR-T cells can also gradually release from scaffold, and especially to boost their infiltration toward the solid tumors, okay, and work together to enhance the treatment efficacy of the CAR-T cells. So if you just purely use the CAR-T cells, and somehow the tumor can sometimes develop the PD-L1 ligand, and then to inhibit this kind of treatment efficacy. Now we can apply the PD-L1 antibody together with the CAR-T cells and to boost the overall efficacy. And actually, a few years ago, we also found that if you locally increase the temperature or locally heat the tumor, and then apply the CAR-T cells, and we found that the infiltration efficiency of the CAR-T cells toward the solid tumor can be also enhanced. And how to heat the tumor, you can apply this kind of photothermal therapy, this kind of ICG molecule that you can encapsulate into such like a microparticles, like a PRGA based, and locally inject to the tumor and shine the light to it, and you can locally increase the temperature. And certainly you can also apply the ultrasound or some other ways to locally increase the temperature. And then together with the infusion of the CAR-T cells, the treatment efficacy can be boosted. Back to the platelets, potentially from the blood bank, or from patient stem cells, or from the certain stem cells, and then you can add a drug to the surface or inside, and then back to the patients. They can target to the specific places, especially associated with the surgical sites or metastasis, and to inhibit the tumor developments. And this is also the major pipeline of our startup company called the capsule, and trying to translate such technology. And we can also genetically engineering the platelets. Platelets, they don't have a nucleus, so you can start with the MK cells and allow it to potentially generate a certain protein, for example, the PD-1 receptor, and then you can add a drug to it, and then infuse back to the patients. Here they can target to the bleeding sites, certainly also associated with the surgery. And once the platelets activate, they can form the platelet-derived microparticles, and then can locally release the drug. And meanwhile, due to the highly expressed the PD-L1 antibody, the PD-1 receptor, and they can interact with the PD-L1 associated with the tumors, and also further enhance the treatment efficacy. And if you run an opposite way, you can also allow the platelets to express the PD-L1, highly express the PD-L1. Then you can just like to inhibit the T cell activity, especially for here, as just like a simple test. We can see here they can especially target to the inflammation sites associated with the pancreas of the new-onset type 1 diabetic mice, and they can even like reverse the blood level. Even such mice blood level went up, and with the injection of such platelets with the highly expressed PD-L1 ligand, and the blood level went down even to a normal range. Actually, 75% of such mice treated by the PD-L1 highly expressed platelets, the blood level become normal. And besides platelets, we are also very interested in other cell types. For example, like fat cell, like adipocytes. You can add a drug to the oil droplet, okay, or lipid droplet of the fat cells, and you can design such a prodrug with the fatty acid, okay, and here's like also doxorubicin, and it's through such linker, especially ROS responsive, and then we can load such drug into the lipid droplets, and especially to leverage the metabolism pathway of such adipocytes in the tumor microenvironment, and we can boost the delivery efficiency of such a prodrug toward the tumor cells and into the tumor cells due to the high level of the ROS. This linker can be easily cleaved, and then to activate the prodrug and to boost the kidney effect. Okay, and we are still working on this and trying to further apply the adipocytes as a carrier to enhance the treatment efficacy toward the solid tumors. And we also develop such like a so-called dead cell-based treatments. And we usually apply the liquid nitrogen to store the cells. And now we can actually get the tumor cells, just like from mice and potentially from patients, and then expand the tumor cells. And then we freeze it through the liquid nitrogen. Then, we can control certain temperature, for example, quickly increase temperature, and then to kill the tumor cells. Okay, we don't want to, to how do you say that, for regenerate such like or like to recover such like tumor cells. Okay, we just don't want to kill them, but still like keep the shape of the such dead cells. You can see like this kind of shape, and certain tumor associated antigen can be still there, and inside you can load the drug, to make it as a carrier, and then deliver to the body. You can gradually release the drug, and meanwhile, due to the tumor associated antigen, they can also serve as the tumor vaccine, and certainly also with such like proteins which can also serve as a targeting ligand toward the certain places, for example, toward the bone marrow or associated with the bone sites, and then to deliver drugs there to boost the treatment. All right, and almost there, and we can also like engineer the hydrogels as a carrier to perform the sustained release of the drug inside the certain place, for example, associated with the primary tumor. Okay, also associated with surgical sites to gradually release the drug. Small molecule drug usually run a bit fast than large molecule like an antibody, right? And sometimes they can work together, even based on the different mass transport, and to synergize the treatments. And not only inject them, you can also spray such like we call the in situ formed hydrogel toward like a bleeding site associated with the surgical site, and to gradually release therapeutic scales like anti-CD47 antibody, also for the cancer immunotherapy, especially to inhibit the tumor regrowth after surgery. All right, and as a brief summary, and currently actually we meet good timing. Okay, I put like 3 pictures here. The first picture is like an X-ray diffraction of the helix structure of the DNA structure. We actually somehow just understand the DNA with such a helix structure within like 70 years. Okay, the past 70 years we understand that. And the second one is like the virus structure, a certain virus, and based on the invention of the TM, then eventually we understood viruses like with such a structure, sometimes with this kind of round spherical structure, sometimes like a worm, sometimes like a different, right? It's like but just like we know this kind of structure, in the past probably let's say around 80 to 90 years, based on the invention of TM. And the third one is like our first picture of the black hole. And It happened like just happened like 2 or 3 years ago. Okay, so that means our understanding about ourselves, about the nature, about the universe is still like a superficial. Okay, and based on this, we can do many things. Okay, actually many exciting things just happened, in the past like 100 years. Okay, and look forward, we can do many even more exciting things. So that's why I always like encourage my students to learn from nature. And because nature is like a very long timeline, okay, and many exciting things, associated with such a very long timeline, okay. And for drug delivery, just enjoy fun and making impacts. And with that, I would like to thank my students. Many of them currently are professors and wonderful collaborators, terrific support from the funding sources. And now our group already moved to Zhejiang University. So welcome to Zhejiang University, actually in a very beautiful city, Hangzhou in China. So thank you so much. Really appreciate your patience.
Thank you for the fantastic talk, Jen. One question is, have you noticed any scarring on the skin post-use?
This is actually
Hi, Jen. Hi, Jen. Somehow we cannot hear you.
Hi, Mehmet, can you hear me?
Yes, yes, we can hear you. So the question
Yeah.
was, have you noticed any scarring on the skin post use of these microneedles?
No, actually, we haven't noticed such scarring. And usually, once you remove the patch, and after 3-4 hours, you can't see, such like how do you say, the holes associated with microneedle is like just recovered. So, but sometimes you can see some like a redness, and different pigs sometimes different, the how do you say it, the situation, yeah.
Another question came in. It was from a therapeutic perspective, the strategies are more on patient specific. What are the strategies that could be undertaken to translate the findings to address patients in a more generalized way in order to enhance the outreach of the therapies to a broad range of patients?
So the question is like to what, to meet the clinical needs?
To generalize, to translate to a group of patients.
Yeah, regarding which technology, the microneedle repatch or?
Microneedle, yeah, microneedle.
Yeah, so regarding design of the microneedle, we needed to, how do you say, to meet the different indications first, like different indications, different situation. We needed to design different microneedles associated with different materials and certain different drug. And sometimes we needed to design some like a dissolvable microneedle, okay, especially for the once or twice this kind of usage. Sometimes for potentially last a longer time, and then you just put the microneedle in and trying to dissolve the microneedle inside and with the formulation leaving inside. And but sometimes, for example, for the insulin delivery, you want to like deliver the insulin like for example every day, and you don't want the material like delivered to the body every day. So then probably needed to develop such microneedles. The materials can't be degraded or can't be dissolved in the body. And so with a certain group, you can develop like one format of the microneedle, okay, like as I said, degradable or dissolvable. But for certain group of with for the the different indication, you need to develop some like a microneedle patch, which can't be degraded in the body. But this is just one example. Okay, so back to my how do you say it, the original thing, I said that every time you needed to look into the clinical need and then to develop this kind of different microneedles to meet the need. Okay.
I see. Another question I have is regarding the delivery from the microneedles. How are the drugs getting diffused? Is the microneedle dissolving? Is the media getting into the microneedles or the body solution?
Can you repeat the question again? It's like you mean the solution?
How are drugs diffusing from the microneedles? Is the microneedle entirely dissolving, or is there another mechanism?
Yes. So, sometimes we call it like a passive, release, and especially, for this kind of water soluble drug, the drug can be, quickly like released from the microneedle patch. Sometimes the solubility is not that good, and then, can potentially last a long time. And meanwhile, as I just, gave the example, which can respond to the glucose level, and then we call it like a controllable release profile. Okay, can respond to the certain like physiological signals, and then like for example, high level biological level, more insulin can be promoted to release. Okay, and of course the most matrix of the polymer is like relatively water soluble. Okay, and so then once the interstitial fluid go inside, interact with the polymer network, and then certainly can also promote the release of drug.
Well, that's all the questions. Thank you for your time. I know it's 1 a.m. in China. I appreciate you're taking the time and giving this seminar, Dr. Gu.
All right. Thank you so much, Mehmet. And thanks so much all. Yeah. Really appreciate it. Take care.